there's a substantial number of genetic changes. And looking specifically at some genes, Vanessa told me that some of these are maintained in coding regions of some oncogenes and others that are carried all the way through from the iPS cells through to the neurons. So I'm not too anxious to have those neurons put back in my head yet. If anybody else wants to try that first, let us know. But I think the field has now gone very broadly with this type of data. And even embryonic stem cells, as soon as they go into culture, start getting genetic changes. What people have seen is first you lose DNA methylation patterns, and then the genome seems to get unregulated and lots of changes happen. So it's very important to understand the nature of those changes. Just taking a cell out of the human body changes its environment, changes its interactions, and therefore changes somehow the regulation of the stability of the genome. If stem cell therapy is really going to be safe and effective, which I hope it will, I think it has more promise for therapy and for understanding basic human biology than almost any other approach that's out there. We need ways to either prevent these changes or to stabilize them. So I'm going to tell you in a few minutes about all our efforts with writing the genetic code. And it turns out you can actually correct these genetic errors in stem cells in culture. And numerous groups have tried this, and it's even nice. You can use one set of the cells as a control and correct the genetic defect in others. And it turns out the various processes for making genetic corrections in the stem cells doesn't cause further genetic change on its own. So all the new methods for synthetic DNA and writing the genetic code tell us that there will be ways to rewrite the genomes and put in essentially error-free or even potentially enhanced stem cells back into people in the future. Number of studies. Here's one just looking on creating a Parkinson's study, correcting the genetic defect in one path of the cells and keeping the others as a control. So it shows you that you can actually correct these mutations. And here's another study where they tracked the mutations and showed that the corrections didn't cause any additional problems. So this is very encouraging, but there's a lot of research going on in just how to stabilize cells and what molecules are needed, what changes chemically that causes these genetic changes, and how, when you take it out of our human body environment, what chemical interactions are lost. So I'm sure those can be mimicked, and we can stabilize them, plus we can correct them. This is a picture of Roger Laskin's robotic instrument that he used for the single-cell genomics. He's been applying this now to single stem cells. So you can actually look at the genome on a single stem cell, or you can actually do gene expression studies on a single human cell. And this is important because scientists are now learning something that shouldn't be a surprise, that our cells are not all identical. They're not all clonal. And when you look at populations, you find a whole range of effects looking at a number of genes. So stem cell genomics, I think, is going to be one of the most important additions to this exciting field of stem cell biology. So we're working on having the San Diego Stem Cell Genome Center, that numerous people here involved in trying to raise funds for that. So on the synthetic cell side, all this work started back in parallel back in 1995 when we sequenced the second genome in history. And it's the genome with the smallest number of genes, 482 protein coding genes and 43 RNA genes. And we just asked a number of questions. How many genes are essential for life? What's the smallest number of genes needed to run a living cell? And then ultimately, could we design and construct a minimal genome, a minimal cell, to try and answer these questions? And as soon as we went down that route, a number of new questions popped up. Would chemistry even permit making large DNA molecules or chromosomes? We could make them, could we activate them? Could we boot them up in a cell? If they're really the software of life, we needed a boot up system. After finishing the human genome, we started in earnest on this, and the we here is Ham Smith, who's been working with me since 1994. He discovered restriction enzymes and got the Nobel Prize for that work in 1978. And Clyde Hutchinson, who basically is an emeritus professor at the University of North Carolina and joined the Institute in earlier last decade to help with this synthetic genome process. And we decided to start with Phi-X174. It's a bacteriophage that kills E. coli. This is the one that Sanger's team that included Clyde Hutchinson sequenced in 1977. And we chose it because it can't tolerate many errors in the genetic code. So we figured it would be a good test for robustness of synthesis. And DNA synthesis is not a very good technique. It's actually very error-prone. It's called an N-1 situation, where the longer you make the piece of DNA, the more errors there are. And so we developed new error correction methods. We started with the sequence in the computer, and within a couple of weeks, we made the entire genome. The exciting phase came next. We injected that into the bacteria E. coli. E. coli recognized our synthetic chemical as normal DNA, started making proteins. The proteins self-assembled and formed the virus. And the virus showed its gratitude by killing the cells that made it, which is how we detect it. So it'll make thousands and thousands of copies of the virus, and the cell will burst open, and then the virus will infect other cells. And this field of bacteriophage is actually coming back into vogue as we eliminate one after another of our antibiotics and have developed a huge range of drug-resistant microbes. Bacteriophage, the viruses that kill bacteria, may come back into being useful. So we call this a situation where the software builds its own hardware. The software was that chemical piece of DNA. The hardware in this case is the virus. But we wanted to make an entire bacterial genome, just not a small virus. But we thought if we could make a lot of these pieces effectively, we could find a way to stitch them together. And that's what we started doing with what looks like a Final Four playoff. We made viral-sized pieces. We put 4 of them together, made these 24,000 base pair pieces. We cloned them in E. coli, grew them up, so we had a lot of DNA. We sequenced it to make sure nothing had altered the DNA sequence, and then assembled those together to make 72,000 base pair pieces. And we worked our way up until we got to molecules the size of 1/4 the genome. And we found that E. coli didn't really love these large foreign DNA pieces. So we looked around for a new system, and we found brewer's yeast actually likes to take up DNA, likes our synthetic DNA, and also has another unique property of using homologous recombination to put pieces together. So all we had to do was put the 4 synthetic quarter molecules into yeast with a synthetic yeast centromere. The centromere on a chromosome is that point at the middle of the Y you see in pictures of chromosomes. And yeast, using its homologous recombination, just assembled all these pieces together essentially instantly. And that gave us the first complete synthetic chromosome that we reported in 2008. And this was of the genome that we did in 1995. And at the time, this was the largest molecule of a defined structure ever synthesized. The team kept working on better advances in the chemistry. Early on, we hired a young postdoc, Dan Gibson, who made a real breakthrough. He took these steps that we were doing one at a time, and he combined them together in one test tube at 50 degrees centigrade. And so it's a single pot reaction. We just throw in these small DNA pieces, drop them in this tube, heat it for a few minutes, and presto, we get assembled pieces of DNA. What that means is it can be automated. So Dan's team at Synthetic Genomics has been automating this, so we go from just this digital world in the computer to making large pieces of DNA At the same time, we were working on the chemistry. We had another team that worked on the biology. It turns out the biology was a little bit harder to solve than the chemistry. But this study that we did in 2007, headed by John Glass on the right and Carol Letigue, a French postdoc, made a real breakthrough. And in this study, we, by changing out the software, converted one species into another. And because of the importance of this to understanding biology and certainly to the future of synthetic species, let me walk you through that because it's very important. So we isolated the chromosome from a species of mycoplasma called M. mycoides. We treated it harshly with enzymes that chew up proteins because we wanted to show that just naked DNA would work for transplantation. We also showed that only supercoiled DNA would work, so you can't have any cuts or nicks in it. We added a couple of gene cassettes, one so we could select for it, an antibiotic selection, and another so it would turn cells bright blue if it was activated. We developed new ways to move these genomes around because DNA, when it's very large, is very brittle, and you can't pipette it in the laboratory. So we developed these tiny little gel blocks, and we move the DNA in and out with electric current, and we can process everything in these gel blocks. And we worked out a way to do genome transplantation of getting that genome into a recipient cell, which is a related species about the same distance we are from mice. So let me show you this very sophisticated movie we have to show you what happened. So we inserted the new genome into the cell. And so now we have a species, the body of one species and two sets of DNA software. So what happened, the new software started being read immediately. Some of the first proteins produced are the restriction